Good day students, welcome to zoology class. Hope all of you are fine. Today in continuation to my presentations on genetics, today is the sixth topic and the topic for today is the DNA fingerprinting. So what is DNA fingerprinting? Actually, DNA fingerprinting can also be called as DNA profiling, DNA testing, DNA typing, or genetic fingerprinting. It means whether we say DNA profiling, testing, typing, or genetic fingerprinting, these are one and the same thing. So today we'll take up different concepts about the DNA fingerprinting. So the process of DNA fingerprinting was developed by Professor Alex Jeffrey at Leicester University in 1984 as a form of genetic analysis, and it was first used in the law courts of England in 1987 to convict a man in a rape case, and it has now been used successfully in many crime and paternity cases worldwide. So the process of DNA fingerprinting, it was started in 1984 and it was developed by Professor Alec Jeffrey in England, and at first it was used in 1987 to convict a man in rape case, but now it has been used successfully in many crime cases or paternity cases worldwide. So what is DNA fingerprinting? Actually, DNA fingerprinting is a way to identify a certain individual rather than simply identifying a species or a particular trait. And the technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. So, DNA fingerprinting is actually a technique to identify a particular individual even of the same species, and this technique can distinguish between individuals of the same species and using only samples of their DNA. So we have to use only the samples of DNA and we can distinguish between two individuals. So the samples of DNA for fingerprinting or for DNA fingerprinting are taken from the blood, hair or saliva, semen, body tissue cells, or DNA samples have been obtained from vaginal cells transferred to the outside of a condom during sexual intercourse. So the samples for DNA fingerprinting can be taken from the blood cells, hair, saliva, semen, body tissue cells, or DNA obtained from the vaginal cells. So these are the different biological samples which are used for the DNA fingerprinting or for the process of the DNA fingerprinting. Now, what are the steps to DNA fingerprinting? Actually, there are 8 steps for DNA fingerprinting, and number 1 step is isolation of DNA. That is, we have to separate the DNA from the sample. And how it is done? The DNA should be recovered from the cells or the tissue, and only a small amount of blood, hair, skin or any other sample is needed to isolate the DNA. So the first step is the isolation of DNA, and it is done by taking only a small amount of the DNA sample. It can be in the form of the blood, hair, skin, semen or vagina or anything else. So the tissue is taken and from the cells DNA is isolated. So here this is the process shown that how the isolation of DNA is done. So this is the first step for the process of the DNA fingerprinting. So now comes the second step. So second step is the digestion of DNA by restriction endonucleases. And the DNA is cut into fragments using restriction enzymes. And each restriction enzyme cuts DNA at a specific basic sequence. So after isolation of the DNA, as we studied in first step, the next step is digestion of DNA. Digestion of DNA means to cut the DNA into fragments. And how to cut the DNA into fragment? It is done by the restriction enzymes. These restriction enzymes, they are also known as the biological scissors. That is, they cut the DNA into fragment, and each restriction enzyme cuts the DNA at a specific base sequence. And what is base sequence? You know that in DNA there are nitrogenous bases. They are adenine, thymine, cytosine and guanine. That is, A is also always bonded with T and C is bonded with G. So this is C and G, A, T, T, A and CG. And so each restriction enzyme, this is a biological scissors, and each restriction enzyme cuts the DNA into fragments at a specific base sequence. So this is the second step. Now what happens that the sections of DNA which are cut by the restriction enzymes, they are the restriction endonucleases. They are known as the restriction fragments. Now we have the fragments of DNA, and this yields thousands of restriction fragments of all the different sizes because of the base sequences being cut may be far apart. Some fragments are long and some fragments are short. So these restriction enzymes, they cut the DNA into unequal segments. Some segments are long and some segments are short because of the base sequences. Because restriction enzymes cut each restriction enzyme cut the base sequences at a particular place. So the commonly used restriction enzymes are HAE third, HINF first, ALU first, etc. So these are the names of the restriction enzymes. And the reaction mixture, it is incubated overnight at 37 degree centigrade. So to cut the DNA into fragments, these restriction enzymes, they are put in the DNA and the reaction mixture is incubated overnight at 37 degree centigrade. So this is the second step. Now comes the third step. In third step, electrophoretic separation of different fragments is done. So what is this? That is, the fragments are separated on the basis of their size using a process called gel electrophoresis, and the DNA fragments are injected into wells and electric current is applied into the gel. So when after isolation of DNA, digestion is done, that is the DNA fragments are formed because of the restriction enzyme. Now next step is to do gel electrophoresis, and because of the gel electrophoresis, these different sized DNA fragments, they are arranged according to the size, and this process is known as the gel electrophoresis. And in this, the DNA fragments, they are injected into wells and electric current is applied along with the gel. So this is the gel and this is the agarose gel, and from here DNA fragments of different size, they are put in this agarose gel well, and this is electric current cathode and anode. So what happens that now DNA is negatively charged, so it is attracted towards the opposite end, that is the positive end of the gel, and the shorter DNA fragments, they move faster than the longer fragments, and DNA is separated on the basis of the size. So this is the process of the gel electrophoresis, that in the agarose gel, the different sized DNA fragments, they are put on it and electric current is passed and DNA is negatively charged, So DNA fragments move towards the positive charge, and the small fragments of DNA, they move faster, and the large fragments, they move slower. So DNA is separated on the agarose gel on the basis of the size of the DNA. Now, the fourth step in DNA fingerprinting is the transfer of DNA on nylon or nitrocellulose membrane. Now, the DNA fragments which are arranged according to the size, they are transferred to a nylon sheet by placing the sheet on the agarose gel and soaking them overnight, and this process is known as the southern blot. So, after isolation, digestion is done, means the DNA fragment is cut into fragments. Then after that, DNA fragments are arranged according to the size by the process known as gel electrophoresis, and now it is a transfer of DNA from the agarose gel sheet to the nylon or the nitrocellulose membrane, and it is done by transferring the DNA fragments to the nylon sheath by placing the sheet on the gel and soaking them overnight, and this process is known as the southern blot. And now next step is probing or probe labeling. Now, after transferring the DNA fragments on the nylon sheath, probing is done. And how it is done? That adding radioactive or colored probes to the nylon sheath, which is complementary to target sequences. And each probe only sticks to one or two specific places of the sheets. So now what happened? Then when probing or the radioactive or colored probes, they are added on the nylon sheath and they attach to the complementary base sequences. Then which radioactive probe we put, we know that sequences, and if there is A, it will attach to the T, and if there is C, it will attach to the G. So this is the probing or the probe labeling. So now next step comes the hybridization, and now labeled probe DNA should be hybridized with the complementary sequences located on the nylon membrane for the detection of the position of the later, and membrane is washed to remove non-specific binding and clearing of the background. As I told you that after probing, the particular base sequences, they attached with the complementary base sequence. If there is adenine, it will attach with the thymine. If there is cytosine, it will attach with the guanine. So now we can see that which sequence is which, and then this membrane is washed to remove the non-specific binding, and then background is cleared. So this is hybridization. Next step is autoradiography, and in this step it is done to detect the sequence in genome bound with the hybridized radioactive probe on the membrane, and technique involves alignment of hybridized membrane with X-ray film, and X-ray film alter its development, shows multiple number of bands that look like barcodes, and they are known as the DNA fingerprints. So in this step of autoradiography, and this material which is prepared earlier in step number 6 after hybridization, that is X-ray is passed through this, and X-ray film alters the development, and there are seen multiple number of bands which look like barcodes, and these are actually the DNA fingerprints. So this is the 7th step in the process of the DNA fingerprinting. Now comes the 8th step, and that is the last step, and this is known as interpretation of the band patterns. Now the band patterns which we get, they are now interpreted. And how they are interpreted? The analysis of band pattern of different individuals is done, then comparison of position of bands is done, and then computer software are also available for the analysis of the DNA fingerprints. So the product which we get, that is known as the DNA fingerprint fingerprint, and it shows certain bands in the form of these bands. They look like barcodes, and these barcodes, they are interpreted. Some computer software also present, and these bands can also be compared by analysis of the band patterns of different individuals. So these are the different steps of the DNA fingerprinting, and as a result we get DNA fingerprint. So this is the diagrammatic representation of all the 8 steps of the DNA fingerprinting. That is, this is the blood sample. From blood sample, we get DNA, and from DNA, electrophoresis is done, gel electrophoresis is done, and the DNA fragments, first they are cut by restriction enzymes. Here they are arranged based on the size by the process of gel electrophoresis. Then DNA probing is done. Then afterwards the radiofilm is passed, and in this way X-ray film is we pass the DNA fingerprint through the X-ray film, and we get this type of the print. This is the DNA fingerprint, or we see here bands in the form of the barcodes, and these barcodes then are analyzed. So these are the different steps in DNA fingerprinting, and as a result we get the DNA fingerprint. Now comes the applications of the DNA fingerprinting. So first application is diagnosis and developing cures for inherited disorders. So how this DNA fingerprinting is used? So DNA fingerprinting, its first application or the first use is diagnosis and developing cures, treatment for inherited disorders. DNA fingerprinting is used to diagnose inherited disorders in both prenatal and newborn babies in hospitals around the world. Then these disorders may include cystic fibrosis, hemophilia, Huntington disease, familial Alzheimer's, sickle cell anemia, thalassemia, and many other diseases are cured because of the DNA fingerprinting. And early detection of these disorders enables the medical staff to prepare themselves and the parents for the proper treatment of the child. And in some programs, genetic counselors, they use DNA fingerprint information to help the prospective parents. So this is the first application of the DNA fingerprinting. Then comes these DNA fingerprinting, the prospective parents, they use DNA fingerprint information. And the second use of the DNA fingerprinting is biological evidence to identify criminals. Criminals can also be detected because of the DNA fingerprinting. Where DNA prints are not available, biological specimens are available like blood or semen stains, hair, or items of clothing at the scene of the crime. Then these items may prove to be valuable sources of DNA of the criminal. So this DNA fingerprinting process, it is very helpful in the biological evidence to identify the criminals. And since the year 1987, innumerable cases have been solved with the help of the DNA fingerprint evidence. So this is very useful. So next is the paternity dispute. So DNA fingerprint is also used to solve the paternity issues. Another important use is paternity dispute solving, and DNA fingerprints in the court system are used to establish paternity in custody and the child support litigation. And in these applications, DNA fingerprints bring an unprecedented, nearly perfect accuracy to the determination of the paternity issues. So this is very useful. And the next application is the personal identification. DNA is the best way to identify a person, as all body tissues and organs of the same person, they contain the same DNA type, and the specimen required also is very small. And in fact, the US Army has been doing DNA fingerprinting of all its soldiers and has a huge data bank. So in US, the army people, they have to do DNA fingerprinting. So the US Army has been doing DNA fingerprinting of all its soldiers, and they have a huge data bank that is DNA fingerprints of all the army persons. So these are the different applications of the DNA fingerprinting. So this is all about. So today you have studied different concepts about the DNA fingerprinting. That is, what is DNA fingerprinting, who invented this, how it is used, and what are the different steps to form the DNA fingerprint, and what are the applications or what are the uses of DNA fingerprinting. So this is all about the DNA fingerprinting. Now you have to go through this presentation, then you have to prepare note for this, and if you find any difficulty while studying this topic, you can discuss those doubts in the virtual class. Thank you.